Hello friends, today I am going to explain about the protein synthesis mechanisms. Protein synthesis means that is the from mRNA to protein will going to be synthesized. Here this is the total last steps of central dogma process. As you can see here in central dogma process, first DNA to mRNA usually synthesized. This process is known as the transcriptions, and from mRNA protein usually synthesized. This process is known as the translation. Protein usually consists of amino acid sequence and DNA mRNA consist of nucleic acid sequence. So genetic information comes from the nucleic acid sequence to amino acid sequence. Today I'll explain about the last part, that is the translations event in prokaryotes. So now before going to the topic, I have to tell about the requirement of protein synthesis. There is a three requirement of protein synthesis, that is the RNA, ribosome and enzymes. Protein synthesis usually occur in both prokaryotes and eukaryotes, and both in both case, that is in case of prokaryotes and eukaryotes, the translations process occur in the cytosol. Eukaryotes usually have that nucleus. So the transcription process, that is the DNA to mRNA synthesis process usually occur in the nucleus, and mRNA to protein synthesis process occur in the cytosol. Now I'll explain about the requirements. There, as you know, three kinds of RNA is present in organisms or inside in cell. That is the mRNA. mRNA means the messenger RNA, rRNA and the tRNA. rRNA is the ribosomal RNA. This RNA is situated in the ribosome, and tRNA, that is the transfer RNA or adapter RNA. This RNA is present within the cell. And next, that is the ribosomes. Here we are talking about the prokaryotic protein synthesis process. So usually two types of ribosome present in the cell. One is the one for the eukaryotes, another for the prokaryotes. Prokaryotic ribosome, usually 70S ribosome, consists of two subunit, and one is the smaller 30S subunit and another is the 50S ribosomal subunit. In case of eukaryotes, that ribosome usually the 80S ribosome. And enzymes is usually performed in the activation process, activations of tRNA process and amino acid process. Enzyme usually participate. So first I will tell about the mRNA. mRNA is the single stranded nucleic acid sequence. As you can see here, that is the single stranded nucleic acid sequence usually coming from the double stranded DNA in case of E. coli. This mRNA usually consists of two parts. One is the coding sequence and another is the non-coding sequence. Coding means they are able to code the polypeptide chain, and non-coding means they are unable to code the polypeptide chain. This coding region is usually started with the initiation codon. Initiation means that codon which is usually able to code the amino acid, and that 3 as you know, 3 codon usually code 1 amino acid. As you can see here, that is the AUG, that is the start codon, is usually present in the mRNA. This AUG usually code 1 amino acids called the methionine, and according to that codon, it will first bind at the mRNA. So this is the things, that is the coding sequence. That means they are able to code 1 amino acid. Now I will tell about the non-coding. Non-coding means they are unable to code any amino acids. This non-coding sequence is usually two types. That, as you know, in mRNA two site is there, that is the 5' site and 3' site. In case 5' site before the initiation codon, that means before the codon where before the initiation codon means from where the initiation process or protein synthesis going to be start. Before that region is present that unable to code any amino acid. This sequence is known as the leader sequence. This sequence unable to code any amino acid, and if that non-coding sequence present at the 3' region, but then it's known as the trailer sequence. If it's present in the 3' regions means after the terminations codon. Terminations means where the protein synthesis will going to be stopped. Suppose this 3 codon is the termination codon, and after that sequence will that non-coding sequence will be present. So this is the they are unable to code. That is the that sequence is known as the trailer sequence. So this is all about the non-coding sequence. They either situated before initiation codon or make and make and situated after termination codon. This is all about the mRNA. And one more things I have to tell, that in case of prokaryotes, that mRNA usually mRNA usually cystone means the coding sequence, so that more than one coding sequence is there in case of prokaryotes. But in case of eukaryotes, that mRNA usually monocystonics means only one coding region usually present. Now I will explain about the rRNA or ribosomal RNA. Ribosomal RNA, the name itself indicating that it's present in the ribosome. There are 3 types of rRNA present in the ribosome. These 3 types are, that is the 5S rRNA, 16S rRNA and 23S rRNA. That 16S rRNA usually present at the smaller subunit. As I told you before, ribosome consists of 2 subunit. One is the smaller subunit and another is the larger subunit. So 16S rRNA usually present at the smaller subunit, and 30S ribosomal subunit is present 50S and other ribosomes, that is the 5S rRNA, other RNA 5S rRNA, 23S rRNA is present at the 50S ribosomal subunit. Now once that mRNA usually synthesized from the double stranded DNA. I will show you in picture. See, this is suppose this is the double stranded DNA, and from double stranded DNA that mRNA usually synthesized. Once it's synthesized, that is the 5' region, that ribosome usually bind at the 5' region. So that mRNA usually carry multiple ribosome binding site. This ribosome binding site is known as the Sine-Dahl-Carnot sequence, and here you can see one of the DNA strands serve as the template strand and another is the non-template strand. That sequence is present in the non-template strand is present in the mRNA. So that this non-template strand is known as the sense strand, and this template strand is known as the nonsense strand. Now ribosome how bind with the mRNA? That as I told you before, 30S ribosomal subunit usually carry that 16S rRNA. This 16S rRNA is complementary to the mRNA or Sine-Dahl-Carnot sequence, so that ribosome usually get attached with the mRNA. Complementary sequence is the 16S rRNA, that is complementary of mRNA, so that ribosome get attached with the site. This is all about the rRNA. Now I will tell about the tRNA. tRNA, that is the transfer RNA. Transfer RNA, as the name itself indicating it will transfer one amino acid, and it serve as adapter. Why it's serve as adapter, I already told you that is mRNA usually the able to code the 3' codon usually code one amino acids, but they do not directly participate for coding program. And here the tRNA usually serve as adapter. I'll show you the picture of tRNA. See, this is the picture of tRNA, and in this picture you can see this is the acceptor arm. Acceptor arm means where the amino acid will going to be incorporated, and this all are the loop like structure. Usually tRNA is a folded single stranded RNA, and here that only nucleotide bases is there. There is no base pair, or there is no hydrogen bond formations. Only single nitrogenous base containing nucleotide, that only phosphodiester backbone is there. So here this portion is usually helps to incorporate, this portions usually accept the amino acids, and this total arm is known as acceptor arm. 4 nucleic acid, 4 nucleotide is there, and here 7 base pairs. That means a double stranded, that is it's single means it's folded structure, so that it create the bond with each other. So 7 bond is usually present in the tRNA. Here you can see this is the acceptor arm, and nucleotide bases, that is the adenine, cytosine, usually ACC bases usually present at the 3' site, and amino acid usually bind at the bind in this site, and but that this site is the anticodon part. Anticodon means the complementary of mRNA. Usually what happened, so many tRNA usually present inside in cell, and as you know, 22 amino acid participate in the protein synthesis mechanisms. At least 32 to 45 in case of prokaryotes, that tRNA will be present, and they will carry 1 amino acids. First tRNA will read what codon is present in the mRNA. As you can see here, this is the start codon which is present in the mRNA, that usually code the methionine amino acid. So tRNA, cytosol tRNA read the amino acid sequence, and according to that tRNA containing tRNA bring the amino acids. And this mRNA, this initiation, this anticodon part, of tRNA usually complementary of the mRNA. So that first they will read the sequence, what sequence is present at the site, and according to that tRNA bring amino acid towards the site. So this is all so that it known as adapter, or it serve as adapter. And apart from that, usually they have a loop and arm form, means arm structure, but that they have that tRNA have D arm, T arm, and that is the anticodon part. Anticodon is the complementary of mRNA. That total process of protein synthesis is usually divided into the 2 parts. One is the hardcore protein synthesis process, and another is the activations process. First, that tRNA and amino acids will get activated. This is the first step of protein synthesis. How they will activate it? First, first amino acids usually bind with the ATP, and this here the enzyme usually participate. The enzyme name is amino acyl tRNA synthase. This amino acyl tRNA synthase usually first helps to attach the ATP with the amino acids and form the amino acyl adenylate complex. This amino acyl adenylate complex method, that means के activated amino acids, means amino acids plus ATP that get activated, and activated form activated, and they generate that amino acyl adenylate complex, that is the activated amino acid. This activated amino acid next bind with the tRNA and generate the amino acyl tRNA. Here in this activated form, enzyme also present, that means के amino acyl tRNA synthesis, as I told you before के amino acyl tRNA synthesis is usually present with this complex. First they will help to attach with the ATP, then within the complex, so within this complex enzymes usually present and helps them to attach with the tRNA. According this attachment, that means keep attachment with the tRNA. According to the attachment with tRNA, this amino acyl tRNA synthesis enzyme is divided into two classes, class 1 and class 2. As you know कि phosphodiester backbone, that ribose sugar is usually associated with the nitrogen base, and at the 3' site of the tRNA, adenine usually present, as I showed you before. That is the first ACC nucleotide bases usually present at the 3'. So first base is the adenine. So adenine associated ribose sugar is there. That ribose sugar have, that is the 2' hydroxyl group. This 2' hydroxyl group create one nucleophilic attack to the amino acyl AMP complex. That means the activated amino acids associated with the AMP, adenosine monophosphate, as I showed you. First it's attached with the adenosine triphosphate and PPI is generally released out, so that here that is the amino acids associated with the AMP complex is there. So that they first create the nucleophilic attack toward the amino acyl AMP complex and create bond with each other, and ultimately generate the amino acyl tRNA in case of class 1. Now I will explain about the class 2 of amino acyl tRNA synthesis. In case of class 2, that ribose sugar, 3' hydroxyl group of ribose sugar usually create the nucleophilic attack towards the amino acyl AMP. So this is the difference between the bond formations. One create bond with the 2' hydroxyl group, another create bond with the 3' hydroxyl group, and according to that they are divided into two classes. So after that nucleophilic attack, they create the bond, that means the amino acids create the bond with the adenine, which is situated at the 3' end of the tRNA, and ultimately generate the amino acyl tRNA. So this is all about the activation process, activations of amino acids as well as activations of tRNA. Now I will explain about the hard code protein synthesis process. This hard code protein synthesis process usually divided into three parts. One is the initiation, elongations and terminations. First I will explain about the initiation process. Before going to the topic, I have to tell about the requirements. So requirements, initiation factor is the major requirement of initiations, that is the IF1, IF2 and IF3. Three initiation factors is present. Apart from that, ribosome, mRNA, tRNA, all are the total requirement of this initiation part. Now I will explain the process. First, ribosome, 30S ribosome. First, 30S ribosomes present in the cytosols, which get attached with the IF3 initiation factor and IF1 initiation factor. This is the stage 1 of initiation process. They get attached. afterward this 30S ribosomal subunit attached with the mRNA. Here that I have to tell, that is the initiation factor 1 usually bind at the A site, and by IF3 is usually bind with the 30S ribosomal site. Total ribosome usually have 3 sites. One is the peptidal site, another is the amino acid site, and in case of 50S ribosomal subunit, another site is there, that is the exist site. In case of peptidal site, first amino acid containing tRNA usually bind, and amino acid site another next amino acid containing tRNA usually bind. So from in the first stage, that is the 30S ribosome associated initiation factor attached with the mRNA. As I told you before, protein synthesis usually start from the start codon or initiation codon. mRNA usually have that initiation codon. In case of prokaryotes, 3 initiation codon is there: AUG, UUG and GUG present in the mRNA. So once it get attached with the 30S ribosomal subunit, the initiation codon usually bind at the P site. So according to that, tRNA bring the amino acid. Next that is the IF1 usually bind at the A site. Once that total initiation process will finish, then only it will allow to incorporate another tRNA containing amino acid to bind at the amino acid site. Until that, it will prevent to bind new amino acid tRNA here in the A site. So it blocked the A site by IF1, and IF3 usually prevent the associations of 50S ribosomal subunit with the 30S ribosomal subunit. It's generally prevent the associations with the larger subunit with the smaller subunit. When it's finished, they finish the initiation process, then only it will allow to bind with the larger subunit. The first 30S ribosomal subunit containing initiation factor bind with the mRNA, and initiation codon situated in the P site. Now I have to tell about the another factor, that is the IF2. IF2 usually carry the amino acid tRNA. First IF2 get attached with the GTP and become activated. Once it's get activated, according to the mRNA sequence, that tRNA first read the sequence, and according to that it gets attached with the according to that it will bring the amino acids, and first that amino acids containing tRNA get attached with the activated initiation factor 2. Once it's bind with the initiation factor 2, it's create the ternary complex. So functions of IF2 usually carry the amino acid tRNA, and tRNA usually read what codon is there in P site or status or what codon is there in the mRNA, and according to that complementary anticodon will be present in the tRNA, and according to that mRNA tRNA usually bring the amino acid. So once that IF2 get activated, it will attach with the amino acid tRNA and bring the amino acid tRNA to the P site. As the initiation codon, in case of initiation codon present is at the P site, so that they will bring the first amino acid at the P site, and AUG is specific for the methionine. In case of prokaryotes, that methionine usually be formylated. In case of eukaryote, methionine usually non-formylated. So that we are talking about the prokaryotic translation process. So the formylated methionine is there, and after reading the sequence, tRNA bring the formylated methionine at the particular P sites. once it's attached with the mRNA, that is the complement, means once codon anticodon create the bond, that times GTP will get hydrolyzed, and once the GTP hydrolyzed, 50S ribosomal subunit will enter at the site, and all the factors IF1, IF2 and IF3 released out, GTP PI released out, and 50S, the larger subunit, bigger subunit, will get associated with the 30S ribosomal subunit. So this is all about the initiation process. Now I will explain about the elongation process. So this is the starting. First, how that first amino acid will coat the site. Now elongations, how they elongate the chain. As like before initiation process, elongations process also require the elongations factor. Here the elongations factor, that is EFTU, EFTS and EFG. EFTU usually that bring one amino acid tRNA. That means after attachment of first tRNA, they will bring one amino acids at the particular site. And EFG, that is the another elongation factor, usually helps to move 3 codon towards the 5' to 3' directions, and ultimately elongates the chain. I'll tell about this is the process about the elongation. So we can divide the total elongations process in two stages. One is the total peptide amino acid associations, and next is the translocations. First the amino acid associations and translocations. So here that amino acids usually after associations of first F met tRNA at the P site, one that EFTU elongations factor bring one amino acids at the A sites, and ultimately elongates the chain, and that EFG usually hydrolyzed EFG associated with the GTP and produce GDP plus PI, and helps to move 3 usually moves ribosomes towards the 5' to 3' directions, and according to that 3 codon will move, 3 codon will come in the one P side and A side. I'll tell in brief in the next part, that is the part 2.